Good day, doctor and everyone. We are Group 3 from the course STKM 2112 Food Microbiology. Today, we will be presenting on the topic microbial pathogens in food. For our presentation, we have selected Escherichia coli, also known as E. coli, as the pathogenic microorganism. My name is Kojashan with metric number A208505. I will begin with a brief introduction of E. coli. E. coli is an important foodborne pathogen and is closely related to food safety and foodborne diseases. Escherichia coli is a gram-negative, rod-shaped bacterium. It is a facultative anaerobe, meaning it can grow with or without oxygen. E. coli is commonly found as normal intestinal flora in humans and animals. Although most strains are harmless, some strains are pathogenic. Escherichia coli is an important foodborne pathogen associated with food contamination, particularly the pathogenic strains enterohemorrhagic E. coli or EHEC and enterotoxigenic E. coli or ETEC. Common sources of contamination include fecal contamination during food handling and processing, as well as the use of contaminated water in food preparation or irrigation. The example of high-risk food includes undercooked beef and mincemeat, raw or unpasteurized milk and dairy products, and fresh vegetables and fruits. The presence of E. coli is strongly associated with poor hygiene and unsafe food handling practices.
And now, I would like to talk about toxin pathogenic metabolite produced by E. coli. As Jasha mentioned before, E. coli is a highly diverse species, and not all the E. coli strains are harmful and pathogenic. Many E. coli strains are part of the normal intestinal flora. However, the foodborne illness is mainly caused by the specific pathogenic phenotypes. These pathogenic E. coli strains cause disease primarily through the production of bacterial toxin. This toxin enable the bacteria to colonize host tissue and disrupt normal cellular process, leading to tissue damage and clinical symptoms. In food microbiology, the most important toxin-producing phenotypes include Shiga toxin-producing E. coli, which is known as the subgroup of enterohemorrhagic E. coli, as well as the enterotoxigenic E. coli, and we will discuss these phenotypes further on the following slides.
For my part of the presentation, I will be discussing the toxins produced by E. coli. The first one is heat-labeled enterotoxin. It is a protein toxin that is inactivated by heat and produced by enterotoxigenic E. coli. In terms of its mechanism, LT binds to GM1 receptors on the surface of intestinal epithelial cells. After binding, the toxin activates the enzyme adenylate cyclase, which increase the level of cyclic AMP or CAMP inside the cell. This increase in CAMP causes excessive secretion of chloride ions and water into the intestinal lumen, while reducing sodium absorption. As a result, this leads to watery diarrhea. Clinically, LT is commonly associated with traveler's diarrhea and does not cause inflammation or damage to the intestinal lining. Next, the heat-stable enterotoxin. It is a small peptide toxin that is resistant to heat and also produced by enterotoxigenic E. coli. The mechanism of ST involves binding to guanylate cyclase receptors on intestinal epithelial cells. This binding increases the level of cyclic GMP or cGMP. The elevated cGMP leads to increased secretion of fluids and electrolytes into the intestine and decreased water absorption, which again results in watery diarrhea. ST-associated diarrhea is especially common in infants and young children and is an important cause of food and waterborne diarrhea disease.
Among the toxin produced by E. coli, Shiga toxin is the most important in terms of foodborne disease severity. It is produced by Shiga toxin producing E. coli, especially enterohemorrhagic E. coli. Shiga toxin is a protein exotoxin, which is cytotoxin, and it is considered a major virulent factor responsible for severe clinical outcomes. For the mechanism of Shiga toxin, the Shiga toxin first binds to the GB3 receptors on intestinal epithelial cells and is then internalized into the host cell through endocytosis. Inside the cell, it damages the ribosome and inhibits the protein synthesis, leading to cell death and intestinal inflammation. Finally, it will lead to the hemorrhagic colitis and hemolytic uremic syndrome.
Next, I will be explaining intrinsic factors affecting toxin production of E. coli. Intrinsic factors are properties within the food itself that influence the growth of bacteria and their abilities to produce toxins. The first factor is water activity. Water activity refers to the amount of free water available in food for microbial growth. E. coli grow best at high water activity, usually between 0.95 and 0.99. At this high water activity, E. coli can multiply rapidly. This is important because toxin production depends on bacterial growth. The more the bacteria multiply, the higher the chance of toxin synthesis. Foods with high water activity include fresh meat, milk, and dairy products. This food contains a lot of moisture, which creates a suitable environment for E. coli to grow and produce toxin. On the other hand, low water activity can limit toxin production. Foods that are dry, salt, or high in sugar have less free water available. This makes it difficult for E. coli to grow. Secondly, E. coli produce toxin most effectively at near neutral pH, usually between 6.0 and 7.5. In highly acidic conditions, such as pH below 4.5, proteins and DNA inside the bacteria can be damaged. For example, the stomach has a very acidic environment, which normally helps to kill the bacteria. However, E. coli has an acid resistance system, also known as a AR system. This system allows E. coli to survive acidic conditions long enough to pass through the stomach. Once the E. coli reaches the intestine, the pH becomes near neutral. In this environment, toxin genes become active. As a result, E. coli can produce Shiga toxin, which cause serious illness such as food poisoning. The third interesting factor is nutrient availability. E. coli produce more toxin in nutrient-rich environment. Nutrients such as carbohydrates, proteins, and minerals provide energy and building block for bacterial metabolism and growth. When the nutrients are abundant, E. coli can grow faster and activate toxin-producing genes more easily. This means toxin production is closely linked to the availability of nutrients. Research has shown that Shiga toxin genes are regulated by nutrient conditions. For example, low iron availability in the intestine can actually trigger increased toxin production in some strains of E. coli. Food that are rich in nutrients include meats, dairy products, and fresh vegetables. If these foods are contaminated or not handled properly, they can support bacterial growth and toxin production. So, nutrient availability is important because nutrient-rich foods can support the bacterial metabolism and enhance the toxin productions.
Next, I will discuss about the extrinsic factor affecting production of E. coli. The first one is temperature. 37 degrees Celsius is the standard for maximizing biomass quickly. For recombinant protein production, lower temperatures are often used to prevent incalcium body, the clumps of misfolded protein, and improve solubility. E. coli grow generally slow, below 10 degrees Celsius and above 45 degrees Celsius. For pH level, the ideal pH for production is usually 7. As E. coli consumes glucose, it releases acetic acid, lowering the pH. If the pH drops below 5, growth generally stops. The third one is gaseous environment. E. coli is a facultative anaerobe, but its production efficiency depends heavily on oxygen. In high-density culture, oxygen becomes the limiting factor. Aerobic respiration produces more ATP than fermentation. The last one is pressure and osmolality. High concentration of salt or sugar in the media increase the osmotic pressure. While E. coli can tolerate some stress, extreme osmolality causes the cell to dehydrate and also known as plasmolysis. Extrinsic addition of compounds like sorbitol or glycine betaine can help the cell survive high pressure environment.
Good evening. My name is Lee Junhua and I will present you about mechanism of transmission of E. coli. E. coli is a bacterium that is commonly found in the gut of humans and warm-blooded animals. Although most strains of E. coli are harmless, some pathogenic strains such as Shiga toxin-producing E. coli can cause foodborne disease. The primary reservoir of pathogenic E. coli is mainly cattle. Other ruminants, such as sheep, goats, and deer, are considered as significant reservoirs too. In addition, pathogenic E. coli has been found in other animals and birds, such as pigs, horses, pets, chicken, and turkeys. The next step is fecal shedding of pathogenic E. coli by animals. The infected animals shed pathogenic E. coli in their feces and contaminate the environment. As a result, this pathogen can pollute manure, soil, and water sources such as wells and water troughs. Farms environments are particularly vulnerable because E. coli persists for extended periods in soil and water. In a contaminated environment, pathogen E. coli can enter the food and water supply through multiple exposure routes. One major exposure pathway is during primary production, where fresh vegetables such as lettuce and spinach may become contaminated when grown in fields exposed to contaminated soil and water. Another important pathway is during the slaughter and processing of meat. The fecal contamination of raw meat can introduce E. coli, and also in ground meat products, the grinding process can spread the bacteria throughout the product and increasing the risk if the meat is undercooked. Not only that, milk and dairy products represent another exposure route when milk is consumed raw or processed without proper pasteurization. Waterborne exposure also plays a significant role when humans ingest contaminated drinking water or consume foods processed with decontaminated water. After the food is contaminated with pathogenic E. coli, the risk of infection escalates during improper food handling and preparation. Cross contamination occurs when raw foods transmit bacteria to ready to eat foods through hands, utensils, or preparation surfaces. And also, insufficient cooking of contaminated vegetables and meats also allow pathogenic E. coli to survive in the food. These transmission pathways result in human exposure. The most common route is oral ingestion of contaminated food or water. Exposure can also occur through direct contact with animals, especially during farms visits or zoo activities. Additionally, person-to-person transmission can occur via the oral fecal route. This all usually occurs in households and childcare environments. Finally, this process leads to human infection with pathogenic E. coli. Some individuals may become asymptomatic carriers who show no symptoms but can still spread the bacteria, while others may develop symptoms, and these symptoms range from mild, such as diarrhea, to severe symptoms, such as abdominal cramps.
So for my part, I would like to explain the mechanism of infection disease for the E. coli. First, the infection begins with the exposure and ingestions. Humans will become infected when they consume the food or water that is contaminated with pathogenic E. coli. The bacteria will survive the acidic environment of the stomach, and the certain strains of E. coli are able to tolerate the gastric acid and successfully reach the intestines. And next, the intestinal colonization will occur, and the E. coli will attach to the intestinal epithelial cells using the adhesins and fibrase, allowing the bacteria to remain in the intestine and establish infections. And this adherence leads to epithelial damage, and some strains such as EPEC and EHEC will form attaching and effacing lesions that can damage the intestinal microvilli and disrupt the normal intestinal space. Next, toxin production occurs. Pathogenic E. coli produce toxins such as enterotoxin and Shiga toxins, and these toxins are responsible for initiating disease by interfering with normal cellular functions. And the fluid disruption and inflammation follow the toxin release, and the enterotoxin will disrupt the ion transport in the intestinal cells, leading to excessive secretion of water and electrolytes into the intestinal lumen. At the same time, the Shiga toxin will damage the intestinal epithelial cells and blood vessels, triggering an inflammatory response. And at last, clinical outcomes and complications develop. This mechanism results in symptoms such as watery or bloody diarrhea, abdominal cramps, fever, and dehydration. And in several cases, the Shiga toxin may enter the bloodstream and causes the severe complications such as hemolytic uremic syndromes, or we call it as HUS.
Alright, now we will move on to the next part of this presentation, which is we will talk about foodborne disease.
Good day, everyone. My name is TING, metric number A211042, and today I will be presenting on foodborne disease caused by Escherichia coli. Now, I will explain hemolytic uremic syndrome, also known as HUS. HUS is a serious condition where small blood vessels become damaged or inflamed. This damage leads to blood clot formation, destruction of red blood cells known as hemolytic anemia, and a reduction in platelet, which is called thrombocytopenia. As a result, HUS can cause acute kidney failure. The population most affected by HUS is young children under 5 years old, as well as people with weakened immune system. The common symptoms include kidney failure, bloody or watery diarrhea, abdominal pain, fever, and vomiting. Patients may also experience extreme tiredness, easy bruising, unusual bleeding from the nose or mouth, and high blood pressure. HUS is usually caused by infection with single toxin producing Escherichia coli through contaminated food such as dairy products and meat, or through close contact with an infected person. In terms of incubation period, HUS typically develop 5 to 10 days after the onset of diarrhea, with a usual range of 2 to 14 days.
Good day, everyone. My name is Cheah. My matrix number is A209351. Now I will proceed and continue with the enterotoxigenic E. coli infection, which also known as ETEC infection. This infection is a type of E. coli that causes common diarrhea in developing countries by producing enterotoxins. This infection especially happened among the children and travelers. The symptoms of this infection include watery diarrhea, stomach cramps, vomit, dehydration, and weaknesses. Some cases even occur fever. The transmission mode or the causes of this infection include the spread in food or water contaminated with the feces. The incubation period of this infection is the symptoms can be seen as soon as 10 hours to 3 days after the infection. The infection also usually occurs less than 5 days, but may also last longer. To prevent ETEC infection, it is encouraged to focus on good hygiene, practice safe food handling method, and be more cautious when traveling to the developing countries.
Next, I will explain enteroinvasive E. coli or EIEC infection. EIEC is a pathogenic strain of E. coli that invades and multiplies within the epithelial cells of the large intestine. It causes a disease that is very similar to shigellosis or dysentery because EIEC uses a similar mechanism to invade and destroy the intestinal cell. The main symptoms include the dysentery-like illness, where patients experience blood and mucus in the stool, along with watery diarrhea, fever, muscle pain, and soft abdominal cramp. Inflammation of the colon may also occur. EIEC infection is commonly caused by consuming contaminated food or water, and it also spreads through the fecal-oral transmission, especially in areas with poor hygiene or through close contact with the infected individuals. The incubation period is usually 1 to 3 days.
Now I will continue to the last part of the infection, which is enteropathogenic E. coli infection, also known as EPEC infection. It is a pathogenic gram-negative bacillus that causes disease in humans, particularly in children under 2 years old. Belongs to the attaching and effacing family to enteric pathogens, which comprises enterohemorrhagic E. coli. The symptoms of this infection include acute diarrhea, mucus production, vomiting, fever, and malaise. The transmission mode or the causes of this infection in three ways. Firstly, occurs via the fecal oral routes. Secondly, it occurs person to person. For example, when there are close contact with the infected person to the uninfected person, especially the unwashed hand after using the toilet, so the infection would be spread around. Lastly, contaminated formines, weaning foods, and infant formula also being the spread of this infection. The incubation period of this infection is between 6 to 48 hours. Lastly, I would like to talk about how to avoid this infection. To inhibit the spread of EPEC infection, we should always focus on good hygiene practices such as wash hands frequently, especially before and after eating, cooking, or using toilet. Also, we should make sure to cook the food thoroughly to avoid the cross-contamination on food. Thank you.